Yes.
Yeah, so greetings to all of our applied mathematics department faculty and students alike. It is a great pleasure to introduce to you our distinguished presenter today, right? This is a new acquaintance, and hopefully this will evolve into more interesting forms of collaboration. This is Michael Sorochan Armstrong, right? If I'm pronouncing correctly. So this is well, I believe it would be only fair to let our presenter introduce himself, right? I would only remark that this is one of the newer connections that we've established, right? Due to thanks to our common friend, right, Ilyakush. So I hope that this collaboration will also grow further, especially given that, as far as I'm concerned, given that the topic of today's seminar is nicely aligned with, well, at least some of the um, faculty's research focus in our department. So without any further ado, let's welcome our presenter, Michael. So the floor is yours, please.
Привіт усім. Мене звати Михайло Сорохан, і я дуже радий спілкуватися трохи українською, але, на жаль, я живу в Іспанії в цьому моменту, і я забув тільки, як говорити українською. Вибачте мене. Ем,
That's fine.
Окей. Доктор Тавров мені сказав, що можна англійською.
Absolutely,
Без
absolutely.
проблеми. Окей. Добре, добре. Well, thank you again for inviting me. I'm very happy to be here, and for personal and professional reasons, I like to try and work with as many Ukrainian institutions as I can. I know from the country of Ukraine, it has a very strong mathematical heritage, and I'm very excited for my I work with in a lot of different capacities. I wear a lot of different hats, but of course, the hat that I believe is the most important is mathematics, because I think if the mathematics is good, all good things come from good mathematical theory, especially in the age of machine learning. So before I get started, I do want to apologize. I just recently switched from LaTeX to LaTeX from Microsoft PowerPoint. So I usually have some really nice acknowledgement slides, but I wanted to thank my supervisor, Jose Camacho, at the University of Granada. He's been a wonderful supervisor, and I'm really proud of the work I do with him in the computational data science lab at the Department of Signal Theory, Telematics, and Communications, which is just sort of a catch-all for a lot of applied math and computer science that we do here at the University of Granada. So thank you again for inviting me, and I apologize for my Ukrainian, but I'm very grateful to be able to speak in English. Okay. So the research I do is part of a European project called Meta-Analyses of Heterogeneous Omics Data. So we have a few key objectives, but the main one is when biomedical scientists, they create a sort of experimental design. They want to often examine the biochemical differences between patients with cancer and people without cancer. And they do this by collecting biomedical samples such as blood, urine, sometimes even spinal fluids, and they put it through these analytical instruments. And from these analytical instruments, it's a multi-stage process, right? We have to analyze the raw signal, and we have to quantify discrete chemical quantities from the raw signal, which is a field unto itself. So I did my PhD in chemometrics, which is the application of math, statistics, computer science to the analysis of chemical data. It's a little bit obscure, but we have some good stuff in there. At least I like to think so. And a lot of my research was focused on extracting discrete chemical information from the raw signal. So for this European project, I still do a little bit of that, but what I'm primarily concerned about is downloading other people's data from the internet for a sort of common disease state like breast cancer, colorectal cancer, even COVID, and analyzing that data within a common subspace. So if you can see my mouse here, you have something, this is sort of the type of data that we usually deal with, right? So along the rows, we have this lovely matrix. Sometimes the matrix is not so lovely. It has some missing values, zeros. So we have samples 1, 2, 3, and each sample is represented by a series of different chemical elements, you know. So this could be vitamin C that you measure in blood or urine or anything like that. This could be urea, all kinds of different stuff. A lot of the time, these modern instruments can analyze thousands, tens of thousands of different chemical constituents in a single run. So within chemometrics, what we typically run into is we have many more variables than samples. So we have to use, you know, either regularization or sort of rank deficient solutions to solve some sort of relationship between the sample characteristics and the observed data. So this is just a little bit of background information about the goals of the project. We get the sort of raw signals. We try to extract the useful information. This is just a variational autoencoder for the sake of example. And then per each sample, we have some experimental factor of interest, and we want to observe it within a sort of common latent space. So the sort of theory is very, very well developed, and for a lot of you, you might not find it particularly interesting. Design of experiments is basically idealized an ANOVA. So this is often when the experimenters, like the clinicians or the chemists, have control over what kind of samples they want to get in order to ask questions about their data, right? So like I said before, here in X, we have our peak table of observations and variables. And under a sort of idealized ANOVA type of relationship, we can factorize this matrix X as a function of the different experimental factors that we might be interested in. So factor A might be whether or not someone has cancer. Factor B might be some other factor like lifestyle. Does somebody do physical exercise? What kind of physical exercise do they do? And this sort of nested factor, this sort of notation we use for nested factors, could indicate where the samples were collected. Were they collected at different hospitals? And would that affect sort of how we infer the disease? And of course, there's also the interacting term between, say, whether or not someone has a disease and their lifestyle factors, X of A, B, and then the unexplained variance. And with the unexplained variance, of course, we can measure it in terms of in a frequentist sort of domain for statistical significance following the Fisher or Snedeker distribution. What we really like to do in our lab, because following the Snedeker distribution requires us to validate ahead of time things like normality, the sort of conditional distribution of the residuals. What we like to do, rather than doing all of this statistical analysis to validate that our assumptions about the data are correct, we often like to do something called permutation testing, where we create the statistic based off of the relationship between our design matrix and the data. Then we permute the data, and then we evaluate the statistic again, and the frequency with which this sort of randomization exceeds the measured statistic, we often say is our p-value. It's very in keeping with the spirit of frequentist statistics. It's the probability of rejecting the null hypothesis. So again, this is just perhaps not very interesting, but I promise it's important. We often encounter these data sets online. They're designed data sets, we like to say. And so for every row of our design matrix, I apologize, it's a D, it should be a C. For every row of our design matrix, say, you know, 1, 0, say that corresponds to disease. 0, 1 corresponds to treatment. -1, -1 corresponds to control. This is following an encoding protocol called sum encoding, and it's designed this way to ensure that each column is orthogonal to each other. Because under an idealized ANOVA scheme, we must have balanced experimental designs. So there must be the same number of participants at each level in the study. And when we don't have balanced experimental designs, we run into trouble. So this kind of trouble we call general linear models. And it's a very, very simple solution to unbalanced ANOVA. All general linear models do is they take this sort of design matrix or coding matrix, and they just create the least squares solution, assuming unbalancedness in the data using the pseudo-inverse. And of course, the pseudo-inverse, just following singular value decomposition, is very easy to calculate, and it accounts for some properties of the experimental design that may not be ideal. So we call this a general linear model. It's different than a generalized linear model. That's something else. That's usually using mixed models and not sort of a sort of classical, not derived from sort of the classical ANOVA analysis in quite the same way. And the interesting thing about these general linear models is that they have a consistent rank, and the rank is decided, the rank of the matrix, the rank of these reconstructions here are decided by the number of levels in the coding matrix. And so when we reconstruct it, according to the solved for regression coefficients as a function of the coding matrix, we get these reconstructions, say X of A, which could be a disease state, that we can represent using something as simple as principal component analysis. And because this represents the average levels, it doesn't represent the error. In order to interpret these models, what we usually like to do is we like to project the error matrix back into the average reconstruction and the sort of calculated, we call them loadings or right eigenvectors or right singular vectors of this sort of representation. And this gives us not only the distribution in principal component space, but it also gives us the uncertainty about the distribution in that principal component space. So that's just a little bit of background information. But the ultimate goal of this project is to find different types of instruments that measure different things, but have a consistent experimental design in common. And these experiments are usually collected from different populations. So if I'm studying the incidence of cancer in Portugal in one study, and I'm studying the incidence of the same cancer in France, they'll come from different populations. If I'm using different instrumentations, the variable space is also different. And it can be very difficult, as you can kind of see here, right? We have this one study, X1, and we have this other study, X2. It can be very difficult to associate X1 to X2 because there's no direct algebraic relationship. And so this is a little bit different because, you know, along the rows, you might have multi-block data where the population is consistent, or along the columns, you might just have replicates in a common experiment. But we're calling this heteromodal data, pardon me. Another common term you might run into is heterogeneous data, but that can refer to a lot of different things. So for our purposes, we're going to call it heteromodal data because the modes of the matrices are not consistent. So,
if you are totally okay with that, because I got used to this practice when people, you know, just interrupt and ask questions along the way.
Of course, I'm totally okay with that.
So just to make sure, so I was just real curious. So when we're talking about heteromodal data, so we have, well, clearly we have different like people, right? But also we have different features. But I was just wondering, so. I would assume that the features, because I'm really struggling to get the problem definition really well. So
no, that's good. That's good.
It cannot be the case, right? That for one sample we only have blood samples or blood measurements, and for the second sample of the different people we only have urine measurements, because that would be totally different. Or is this exactly what you're talking about? That for let's say Portuguese sample, we have absolutely different than we have for French. But is it? Well, of course you're going to talk about this, right? In this presentation. But is it even possible to try to measure these things if they're not even on the same? Well, not even. Well, they come from absolutely different domains, because here you measure blood and then you measure like pressure or something like that. Does it really make much sense? Because to me, it sounds like a very bold endeavor to try to combine the data sets that come from very different, not only populations, but also different feature sets.
I've been asking myself that question every single day. It is indeed very difficult to do. I'll show you in the next couple of slides that the algebraic relationship is actually quite easy. It's actually the easiest part of this. But determining whether or not these two different experiments from completely different domains have anything in common, that's the very tricky part. And that's where it gets very complicated. The mathematics I hope are fairly intuitive, but if they're not, I'm more than happy to talk about it. Just slow down a little bit because it is a little bit maybe not so intuitive because I really enjoy working with linear models because for this type of data we really like to make sure that we can interpret the models, like what say metabolites are strongly correlated with the presence or absence of a disease state, because that kind of information can tell us avenues for possible treatment or diagnostics. If we know exactly what molecules are correlated with its disease state, we can work backwards to the actual biochemical pathways and actually explore potentially what's going down, what's going wrong with the body at a particular point in time. But you're absolutely right. It is a very challenging thing to do. I got some feedback for the grant proposal, and some people liked it, some people didn't like it. A lot of people said it was too ambitious, but we've done our best, and I'm fairly happy with the results, and I hope it's convincing. If it's not, that's okay. I can take pretty harsh feedback. But no, that's a really good question. And of course, anyone, if you want to ask a question, please go ahead. I really don't mind at all. I think that's nice. It's facilitates scientific discussion. But we're all on the same page, right? This is kind of an ambitious task, right? We have data set one, data set two. On paper, they have absolutely nothing in common with each other. But I hope that the sort of discussion I had about the rank of the matrix might lead some insight into the next thing we're going to talk about. So, with things like PCA, a lot of types of multivariate statistical analysis, we rely on the calculation of a covariance matrix, right? But if there's no common samples, no common variables in common with each other, then just by taking the inner products, we can't calculate a covariance matrix very easily. And there are a lot of nonlinear ways of associating this data with each other, of course, but we don't like nonlinear methods in the field I work with as much because they're harder to interpret. And that, of course, touches upon things like deep learning and stuff like that, which are also highly nonlinear models, also very difficult to interpret. So we're working with sort of a limited tool set here. We have very high expectations for what we can do with the data, and we're limited in terms of the tools we can use to solve them. Okay, so like I said before, we have a factorization of a experimental factor of interest. Say it's cancer. Does someone have cancer? What stage do they have? That sort of thing. It can be reconstructed according to the design matrix and the calculated coefficients through the general linear model that I discussed earlier, and there will be some error associated with it that accounts for variability of individuals within group variability, that sort of thing. So by observing that each of these matrices, if they have a common experimental design, so if X1 and X2 have a common experimental design, we can factorize these matrices just using principal components analysis. And using a very simple mathematical step, we find that we can actually align matrix 1 to matrix 2 using orthogonal Procrustes analysis. So all that does is it finds an orthonormal basis that rotates the samples in experiment, in experiment 1, the scores of these samples, to the samples in experiment 2. And then we can even sort of project the reconstructed experiment 2 as a function of experiment 1 using the error and the loadings or the right singular values, pardon me, and then the sort of orthogonal subspace that we've calculated, this orthogonal rotation matrix. So again, this step is called orthogonal Procrustes analysis, and it's very common in information fusion, where we're trying to relate one matrix to another. The problem is that we have no common dimensions. Now, I have to specify something because I said earlier that there may be different numbers of participants, right? And this only sort of accounts for the sort of column space, not necessarily the row space. But keep in mind that each one of these, this sort of reconstruction, according to the experimental design, has the averages, and the averages are not unique. So the averages are degenerate, and because they're degenerate, we don't run into any numerical problems. And we can calculate the subspace without necessarily needing to have different numbers of samples. We can calculate the subspace as though we were looking at the same number of samples. So what does this look like, right? So I talked about experimental levels. This is finally perhaps putting a picture to it. So I am trying to fit the filled dots to the hollow dots. And these come from two different experimental data sets with absolutely nothing in common with each other. They have independent samples and they have independent variables. These were synthetically generated. And I know that there should be some relationship between these two data sets because I've sampled them from the same larger matrix along the block diagonal, like I talked about before. So using this type of analysis, it's very easy just by looking at the averages to calculate the optimal rotation to relate data set 1 to data set 2. And as you can see, it should be fairly convincing, right? They span a very similar subspace. And from this information, we can work backwards and calculate, say, the right eigenvectors or the right singular vectors to find out sort of what variables are responsible for this sort of reconstructed data set. Okay, so I hope that was somewhat convincing, but we run into this a lot with machine learning. Just because you can doesn't mean you should. I mean, you can make a model, a deep learning model, for instance, that can fit anything. The problem is whether or not it's generalizable. And even for simpler models like what we're doing in statistical learning, we still run into this problem, but we're more motivated by theoretical considerations. For example, is dataset 1 from the same distribution as dataset 2? And we're not even really working with the datasets themselves, we're just working with the principal component scores. And the principal component scores famously have an ambiguity. So it's called a sign ambiguity. So if I multiply both the left and the right eigenvectors by a scalar multiple of -1, I get exactly the same result. I get exactly the same reconstruction. This is fairly well known. You can, if you don't believe me, you can just, on paper, just multiply, -1 times -1 for any type of eigenvector decomposition, and you should get the same answer because the -1 times -1 is +1. That's just an intuitive way of looking at it. But ultimately what we get with these design matrices is that we get this additional ambiguity. It's a permutation ambiguity. So what the principal component space looks at is the variance between levels 1 and 2 and 1 and 3, or however many levels you have in your sort of covariance structures that are spanned by the coefficients that you've calculated as part of your general linear model. And so you might see the same structure in your data in principal component 1 in dataset 1 as you do in principal component 2 of dataset 2. So I know it's a little bit abstract, but I'll show you a visualization in the next slide. So what I have here is I have some distribution P of x1 times Q of x2. So these are representing my two different data sets. And in some sort of principal component subspace, they may have different sort of directions in the data. Here is x1, here is x2. As I mentioned before, I'm trying to fit x1 to x2. And as I mentioned before, I can easily do that using an orthogonal Procrustes type of analysis. I can fit x1 perfectly on x2, as you see in this blue sort of ellipsoid right here. Now, if I have a restriction in this transformation, if I'm doing a sign permutation, which accounts for all the ambiguities of comparing two different principal component spaces, you can see it's not a perfect fit, right? Because I can only flip this yellow ellipsoid. Now, because I can only flip this yellow ellipsoid in a way that accounts for the ambiguities in the two principal component solutions, what I can do is I can measure the best possible fit, which is this orthogonal Procrustes analysis, over the nominal fit, which accounts for this ambiguity, which is the signed permutation type of analysis. And so the justification for this is the overlap of the two different Gaussian distributions. Like I said before, what we're looking at is the difference between dataset 1, dataset 2 as a function of some rotation matrix versus dataset 1, dataset 2 versus a function of some signed permutation matrix. The permutation matrix is calculated from the optimum rotation matrix, and I'm calling it the next nearest sign permutation matrix. So if I look at the overlap between these two distributions, just integrating over x2, which is the data set we're interested in fitting, what I find is a very sort of convenient identity. If we neglect the sort of normalization constant right here, we find that the proportion of overlap is equal to matrix x1 times the difference between the rotation and the next nearest sign permutation matrix times this precision matrix, or the inverse of the covariant structures themselves. The covariant structures being the difference here, so the residuals, not necessarily the matrices themselves, but the residuals. So it's a little bit abstract. So in order to test for the relationship, if dataset 1 can truly be said to come from the same distribution that we're interested in, even though it comes from a different population, different variables, different measurements, what this suggests to us in principal component space is that the difference between this next nearest sign permutation matrix and the rotation matrix is in some way proportional to the overlap between these two distributions in principal component space. So how do we test for this? The sort of parametric test is possible. It's actually a type of Bessel function. But because I work in a computer science department and because, generative AI is a really big thing right now, we thought we'd opt for permutation testing in order to establish the significance from a frequentist point of view. So if we get a very, very low p-value, then we say that the relationship is significant. If we don't get a low p-value, we say the relationship is not significant in sort of colloquial terms. So starting off with, dataset 1, dataset 2, the common covariance, the common design matrices, what we do on the left-hand side of this algorithm is we calculate this nominal relationship between the principal component scores. And then on the right-hand side of this algorithm description, we try and test that by scrambling the data and then trying to reevaluate the relationship. So this is a bit of an older version of an algorithm we're working on and we're continually improving. Especially both in regards to how we simulate the data, we create the statistical simulations, and I understand it's probably a little bit complicated, but in principle, there should be some familiar friends here. We calculate the coefficients, we calculate the reconstructions here, we decompose the reconstructions, we calculate the scores. This is just to improve the condition of the matrices. It's a Varimax rotation. And then we sort of calculate these unique experimental levels, perform the orthogonal, recurse these analysis, and then measure this test statistic, where we have the factor of interest and the rotation between these two different data sets should nominally be quite close to this next nearest sign permutation matrix that accounts for these ambiguities in the principal component decomposition. All we're doing here is we're just scrambling the data using permutation of the rows and permutation of the columns, and we recalculate the test statistic, and then we measure the frequency at which the test statistic in this case is actually larger than the test statistic that we should expect to see. Because if the data sampled from the same distribution, the test statistic should be small. And if the permuted data exceeds that test statistic frequently, then we should see a high p-value that would tell us that these data aren't sampled from the same distribution. So this is sort of a work in progress, but you can see when we generate data sets synthetically with a common experimental design separately, basically the significance cutoff, as we modify the significance cutoff, the number of data sets that we report as significant follows a sort of linear trend, which tells us that in the null case, where the data sets don't have any significant relationship, that it follows a uniform distribution. And then this alternative distribution where the data sets come from the same distribution that are block diagonally sampled, we look at the proportion rejected. And so for some, classical alpha significance cutoff of 0.05, we see that the power, or 1 minus the false discovery rate, is 0.8. The statistical power of this test looks to be pretty good. It's not perfect, but it's pretty good considering the sort of magnitude of our problem. So what does this look like with real experimental data? So we find data online from 2 different studies, both studying breast cancer. So this experimental design has 3 levels. Level 0, control. Level 1, stages 1 and 2. And level 2, stages 3 and 4. So these are two ANOVA-like tables. In the first column, we have the mean, diagnosis, our experimental factor of interest, and the residuals. It's not a particularly rich experimental design. We don't have a lot of factors in common. We just have this diagnosis, whether or not the individuals in this data set have cancer, and has the corresponding degrees of freedom, corresponding F value, and then the permutational test statistic for this data set is low. It's significant, appears to be significant. Same thing with this other data set. Same experimental design, F value, Snedeker value, Fisher value. The permutational test statistic, also very low. Now, when we try and measure whether or not it's from the same distribution, we get similarly a very low p-value. So across 1,000 permutations of the actual data, when we're trying to examine whether the subspaces are comparable, the distribution of the data is comparable, we also get a low p-value, which suggests to us, based off of the test statistic that we proposed, that the data sets are sampled from the same distribution. So just in conclusion, thank you very much for your patience. I know it's kind of out there. It might not necessarily be in your lane in terms of being applied mathematicians, but I really, really appreciate your attention. So what we were able to accomplish is in this sort of heterogeneous, heteromodal data space, we have a pairwise statistical comparison. We have visualization and interpretation in an ASCII-like framework. This is ANOVA simultaneous component analysis. This is sort of the theory that we derive this sort of test from. I'm sorry I did not talk about it a little bit earlier. All ASCII is, ANOVA simultaneous component analysis, is taking this general linear model or ANOVA model in cases where the experimental designs are balanced, and applying principal component analysis or simultaneous component analysis when you have more than one different experimental factor, and using that to interpret and visualize the different levels when you have a lot of different variables that you'd like to visualize. So a limitation of this project is that common experimental designs are very rare, but this is a good step. And of course, peak tables themselves are often polluted with zeros, missing data, because the instrumentation itself is very sensitive and prone to malfunctioning. So all of these things represent a challenge for this type of research going forward, but we've made a first step at least. So I swear I'm not making all of this up. These are some very, very important references that have motivated this type of research, starting with Marty J. Anderson. She's an American statistician based in New Zealand. She proposed permutation testing via the Permanova as a way of handling ANOVA type problems with non-normally distributed residuals. This is a very famous paper in my field, at least. Of course, Rasmus Bro and Tamara Bulda are very famous mathematicians. Rasmus Bro is a chemometrician, but he works in a lot of areas. This is something they've done with principal component analysis, singular value decomposition. This is where I got the sign ambiguity from. And of course, ANOVA simultaneous component analysis. This is a common tool in bioinformatics, chemometrics. It's the sort of theory that I derived this tool from. It was proposed by Ahe Smilder, an econometrician and bioinformatician who recently retired from the University of Amsterdam, and his PhD student, Jeroen Janssen. So if you're interested in this sort of thing, ANOVA simultaneous component analysis is a good place to start. And then the extension of ANOVA simultaneous component analysis to unbalanced experimental designs was called ASCA Plus. And yeah, that's all I got. So again, I'd just like to thank my postdoctoral supervisor, Jose Camacho, and the Computational Data Science Lab. I'm so sorry for the lack of an attribution slide, and of course, the government of Spain and the European Union for funding Canadian.
Yes, well, I believe the Europeans are only too happy to fund the Canadians, and also I believe all the Americans who would like to flee the
Yes.
US to Europe and miss this political turmoil that we're enduring. Yeah, thank you so much. It's been quite interesting. I must say that so me myself, and I'm teaching the data analysis course for our upper intermediate undergrads. I always, sort of like gloss over the ANOVA because I always tell them, well, of course, there is such thing as ANOVA, but I mean, there is nothing that ANOVA can do that regression analysis cannot. So my question to you was, or would be rather, so here it's everything is based, right, basically on ANOVA. But can this be can this kind of research can it be like well um, can you use for this kind of research just regular linear regression or any modifications of that? You mentioned at the beginning that in some cases you have well maybe not in this particular empirical illustration, but in some cases you can have much or many more columns that you have observations, right? For that you could probably use less or something like that. Didn't you try to experiment with that kind of analysis? What is so special about ANOVA? Maybe I will teach my students as well.
No, that's a really great question. And this sort of general linear model, the generalization of ANOVA, is a regression. It's ultimately just a regression. It's just a question of how you factor the different variables, right? So if you factor them correctly, the difference between, as you said, the difference between an ANOVA and a general linear model, in cases where the experimental design is balanced, is indistinguishable. They give equivalent solutions. So ANOVA, it calculates the mean sort of manually. Do you have your mean values, if you're comparing the difference between two means, you have your within group variance, your between group variance.
Yeah.
Ultimately, it's the same thing as a regression and its associated error, right? You have your between group variance as the expected values, and you have your within group variance as the error. And just that ratio is the test statistic that you use. So absolutely right. I mean, like you can just use a regression, and because I'm lazy, I'm a lazy computer programmer. It's just easier for me to take this design matrix, regress it against the data I'm interested in. Also, because in that case, there's no difference between a univariate ANOVA and a multivariate ANOVA because it's just matrix multiplication.
Right.
So, it also, to me, is more intuitive because it connects to just linear algebra, which gives me all these nice properties that I can work with, right? If I'm just looking at the sort of these parametric ANOVA equations, I'm not quite as happy.
And also, you mentioned several times. I would even go as far as to say that you were especially careful to stress that in a frequentist setting. So which of course hints to the idea that well, there is a Bayesian setting, right? So the question is, did you, or maybe in your research lab or whatever, do you have like any like threads of research where you actually use Bayesian statistics to do this kind of analysis, or only frequentist?
We're huge fans of Bayesian analysis. We really are. But in this type of research, what we usually don't have are priors, right? And of course, you can
Right.
estimate soft priors. You can sort of estimate them numerically. But because we have all this computational power, it's easier for us to, just simulate a null distribution, even if the data isn't perfect. And sort of what we're doing in our lab is really exploring what we can do perhaps with older statistics and more computational power. But I know in a lot of other labs they do something similar with estimating the priors for like a Bayesian regression and stuff like that. But just in terms of what we're doing, we don't typically work with Bayesian type analysis, but it's really interesting because we do respect it. I don't want to say that we don't respect it because we do a lot of power analyses as well. So once we get our p-value, how confident are we that this p-value is what we're looking for? And that's often what Bayesian type analyses are better at, right? But what a lot of the literature I've read recently has shown that if you do a power analysis and you get sort of things like effect size and stuff like that, you're learning a lot of the same things as you would in a Bayesian type analysis where you do know your priors. It's just a bit more work. So it would be something very interesting to explore because I do like a nice Bayesian regression, but it's
Those ones are
it's
really interesting.
yeah.
Yeah, you see the Bayesian kind of analysis always, it's very close to my heart and has always been, right. So I was converted to this kind of statistics actually at UC Berkeley when the professor said that, well, I mean, everyone should be Bayesian, but unfortunately, the frequentist paradigm has, permitted so much. But yeah, yeah. Thanks. That's most interesting. Uh, Maybe other colleagues want to ask their questions. Please go ahead. Do not be shy.
I'm not a specialist in this particular field, yes, but some very general question, very. Your model is linear. Why it is linear? What can be done to make it non-linear, and whether it has sense to apply some non-linearity in models, some linearization maybe, some very general hints for me, please say.
Actually, that's a really great question, and I kind of brushed up upon it. So I mentioned a bit earlier that my background is in chemometrics, so we're actually quite dogmatic about linear models, linear functions, and we get very upset when we see any kind of nonlinearities. So there's a good reason for this because these types of instruments have a linear relationship between the quantity of sample that they measure and the instrument response. And if there's any sort of non-linearity between that relationship, then we get very upset. And there are ways of correcting for it, using nonlinear methods, nonlinear regressions and stuff like that. But in general, we do like to assume that every one of these columns is some linear function. And if it's not, we get upset. So all of these linear models are based from the actual instrumentation that are used to analyze these types of data. And something I regret I didn't have time to show because I really do appreciate your patience. But something that we do in the computational data science lab is we do sort of rather than making the models nonlinear, we make the basis function nonlinear. So for example, by using Fourier series, if we have a continuous function and there's some drift in it, why don't we just analyze the complex coefficients of a Fourier decomposition? So that's a really good question. Why do I like linear models? Why is all of my research based on linear models? The answer is it's derived from knowledge of what kind of data we're working on. So I'll just say domain knowledge. Thank you very much. That's a really great question.
It is not intentionally a very good question.
No. Well, intentional or not, it was really good, insightful.
Yeah. So yeah. Oh, yes. Go ahead. Sorry, I'm in no position to interrupt you, of course. Igor Vladimirovich, go ahead.
Yes. When you verify your model, you need to use some data. Some of them are generated by you, yes, is artificial data. And which law of distribution that you use normal when you generate your data? You use normal distribution or maybe Weibull distribution or uniform. Which kind of distribution you used to verify your model?
Another really good question. So we can do a number of different distributions. So for this one, we're talking about this sort of analysis where we generate thousands of data sets and we measure how well our algorithm performs relative to when we generate them independently versus when we generate them with some known dependence on each other. So of course, we can modify what distributions we're looking at. But for this one, ultimately is a normal distribution, a multivariate
Always
normal.
normal.
Always normal in this
From
case because
mechanical words, we very often used Weibull distribution, Weibull. It's very popular in mechanics, so I think maybe someone else uses distribution or
no, and that's a good point. I mean, like I do make some assumption about the normality of my data because we have to randomly simulate the experimental levels and we have to randomly generate noise around those experimental levels. So we sometimes we can change what kind of random numbers we use, of course. But the nice thing is, and this is something we'll have to verify when the paper's finished. But you know, when we're looking at stuff like this, we have to verify that it's robust against all kinds of different types of distributions. But another reason that we use permutation testing, and this isn't something that I discussed a lot, but I sort of made an offhand joke that we're a computer science department and we like to use the computer to do some of the work. But oh, lots of questions. Oh, shoot. Sorry, but with the permutation testing has been proven to be more robust against non-normality. So if the data is not normal, it's more or less okay most of the time. But of course, you have to verify that. Sorry. Other questions?
You see, the more you respond, the more questions we get. But of course, this is just a correlation. There is nothing causal about this. So,
See what the questions are.
Vladislav, you wanted to ask a question, I guess.
Yeah. Hello, I'm Vladislav. Sorry. Can you hear me?
Yes, I can hear you
Yeah.
just fine.
Yeah.
Thank you, Vladislav.
Thank you. So I'm Vladislav. I'm PhD student here. So I wanted to ask maybe a step back. So it's really a correlation. It's not related to this answer. But to have a step back, I wanted to return to the kind of problem statement and the objectives.
Sure. I'll just go.
Yeah. So is it?
Yeah.
So do I understand correctly that why we do this is that we basically say that despite those experiments having different features, they still share some kind of same general population that we actually want to estimate and not the samples. Because in theory, right, we can rotate matrices and align any kinds of people or any kinds of features about anything if we don't assume neither features nor the number of participants. Or why are we exactly trying to align them?
No, that's a really, really good philosophical question. And it's a question I had to think about when I was applying for funding. But ultimately, like what we're thinking of is so when this type of research is typically called metabolomics, genomics, proteomics, lipidomics. What that means is that different scientists, different doctors, different statisticians, they're looking at different domains within the body. So with metabolomics, we're looking at blood, urine, we're looking at these types of chemicals that are generated in the human body through biological processes. So philosophically, there should be some correlation between one domain, say if I'm looking at proteins in the body, which generate these sort of smaller molecules, there should be some correlation between what's going on causation really, what's going on with the proteome, the proteins, versus what's going on with the metabolites. Because we know a priori that one causes the other. So when we're looking at these different experimental levels, when we're trying to find correlations with them, the trouble is that some data is really good, some data is really powerful, gives us a lot of insight, and some data isn't. So we need to be able to distinguish when we're looking through hundreds of data sets online, are we converging to something useful, informative about the underlying physiology of the human body? So the sample distribution that we're looking at is actually going to be are we looking at the same thing or not physiologically? I hope that answers your question.
So thank you for the answer, but just to clarify it on the spot. So is it correct that we're looking basically on the same people sick with the same cancer, for example, or are we trying to look in general?
Yeah, so the sort of domain that I've been looking at throughout this presentation is I've been taking X1 and I've been trying to fit it to X2. So what we're doing is we're actually generating the same people in this population, X1, as a function of the variables in x2. So I didn't really get a chance, or maybe I should have spent more time maybe showing some of the mathematics behind how that's possible. But I do think maybe, yeah, something there, right? So these second scores right here, T2, that's sort of what we're looking at here. So it's the same population, just expressed differently. But alternatively, we could look at a different population expressed the same. So we could look for x2, the population of x2, as a function of x1. So,
Okay, I see. Thank you.
no, it's a great question. Thank you very much.
Yes, Anton, please go ahead.
Yes, so first of all, thank you very much for the great presentation. Again, I'm
Yes, you
not an expert in this field either, so I'd like to ask more like general practical question. So because this model is linear and it's regression, how do you usually scale this model in the production for your scenario? And how do you usually combat the scenarios of underfitting and overfitting for your data?
Great question. Great question. So I'm just going to go back down to the practical demonstration towards the end here, because this gives you kind of an idea of a workflow. So when we only have, so I'll start with the standardization versus mean centering. So with principal components analysis or singular vector decomposition used for the purposes of data analysis, which is almost always principal component analysis because there's a relationship between the two, as I'm sure everyone here knows. And if not, that's okay. It's kind of might be an obscure fact for fans of linear models such as myself. But it assumes that the mean is zero across all columns, basically. Usually that's the most basic kind of pre-treatment that we can do for the data. And then depending on how well the data is behaved, we can scale the model differently. So we can do what we call in the business, in chemometrics, we call it auto-scaling. In other fields, you'll call it standardization. So we'll mean center and standardize. And if the data is well behaved, usually that's all we need to do. If there's a lot of outliers, we might need to sort of scale the data, say using log scaling, for instance, just to give some sense of normality in the data, because ultimately any of these models do assume some sort of Gaussian distribution of the residuals, not the data itself, but of course the residuals should be nominally Gaussian. And we kind of see it right here when I sort of specify the model parameters. I'm assuming that my distribution of residuals is Gaussian. In terms of overfitting, when we're building an ANOVA model, when we just have one factor, it's very easy. If we're overfitting the regression, then when we break the relationship through permutation testing between the underlying data and this experimental design matrix, if we break it and we break it and we break it and remeasure that relationship and it never or rarely exceeds the sort of nominal test statistic that we used earlier, then we in general say it's not overfitting. But for more complicated N-way ANOVA models, say if we have diagnosis, lifestyle, all of these different things, right? It becomes a little bit more complicated because what if the factor isn't significant? How do you handle that insignificant factor? If I have 12 different factors, they're each going to explain there's going to be less and less and less unexplained variance in the model. So the denominator for a test statistic shrinks. And so it could be that everything looks significant if we just test for everything all at once. But what we usually do is we do model trimming. So we will sort of test for everything, disregard the insignificant factors, put them into the residual matrix, and then test again. And if these factors that we're interested in survive model trimming, that's generally a responsible thing to do for these types of ANOVA analyses. And I know ANOVA kind of, seems to have been superseded by a lot of more modern methods because it's been around for a while. But the nice thing about ANOVA is it's been studied to death, and it actually continues to be studied. And there are some, best practices for ANOVA that we're still working on today. My supervisor, Jose Camacho, he's actually an expert in experimental design, ANOVA, these types of things, and he's always finding new ways to sort of validate his assumptions about the model, new ways to test if your ANOVA model is any good. And I know a lot of people are moving towards Bayesian regressions, but we can do a lot of cool stuff with just general linear models and ANOVA, such as these lovely pictures I've drawn for you today. So I do still think there's some value, but I understand that Bayesian statistics is a very popular avenue of research. I went to a statistics conference in July, and we were, I guess, sort of the only orthodox frequentists that were left. So it was interesting.
Well, that's actually extremely good, extremely uh, nice for me to hear because it was sort of like on my watch when the business started to get like more recognition over the years because when I first heard about it at Kiev School of Economics of all places, it was like presented with the sort of esoteric regression called Bayesian regression, but now of course it's not esoteric anymore. Right? Yes, that's quite interesting. Quite interesting. Do we have any questions? Do we have any questions from the audience?
Thank you very much for that.
No, thank you, Anton and Vladislav. Very nice questions, and everybody else. Sorry, everyone, thank you so much for your questions. I'm really happy that it was presented in a way that was engaging. First time, I went over this, I wasn't so sure, so it's very nice.
Yeah. So thank you. Yes. So I believe that no one else wants to ask any questions, right? Oh, Ilya, you want to maybe not ask a question, but maybe comment on something? You're muted. You're muted. Ilya, unless you unmute your microphone, we cannot hear you. Oops.
I feel terrible because Ilya has helped me with my Ukrainian. Ilya.
Yes. No.
Hope you hear me.
Yes.
Hi, Ilya.
Hi. Nice to see you, Michael. Great
Nice to see you too,
you're
Ilya.
so active and help us and show your results.
I also I owe Ilya a lot of thanks because he arranged this talk with Dr. Tavrov, and I also need to thank Ilya because he helped me a lot with my Ukrainian for the last several years, and I've been out of practice for a few years, but I hope I did not dishonor Ilya's teaching too much.
And after your words, now you have to practice. Now.
Yes. Але я сказав, що небагато українців в Гранаді, тільки іспанців.
Well, I find it hard to do. Oh, yes, go ahead.
Now you can communicate with many Ukrainians about applied math stuff. So you're welcome.
Yes,
Thank you very much, Ilya.
Yes. We will do it on a rolling basis, right? I will appoint people responsible for communication. No, thanks so much. Of course, thanks to Ilya for making this connection happen, right? And hopefully we will try to develop to grow, right? To make it better and to find some areas or like points of connection that because maybe Igor Volodovich is an expert in mechanics and geometric modeling. Maybe I'm more leaning towards like Bayesian statistics and stuff like that. But and Anton, for instance, is a guru on optimization and stuff, right? But here at the department we have other people, right, who are more or less related to like pure statistics, right? So
I also do other stuff. I also do other stuff. So it's not just this. I'm sure we can find lots of stuff. I mean, you can. I spelled my name with traditional Ukrainian transliteration into English, but you can just Google Michael Sorochan Armstrong, and if you want to make fun of my papers or or if you're interested in them, that's also very nice.
Sure. That would be very good because you see one of the advantages and also disadvantages, right? At the same time of heading the department of applied math is that you have lots of very different people, right? Because if it were, for instance, like the department, I don't know, differential equations, I mean, most of them will be just experts on differential equations, right? But here
Of
in the
course,
applied
of
maths,
course.
yeah, every single person is an expert on a totally different fields, right? Ranging from machine learning to reinforcement learning to partial differential equations to splines to whatever stuff, right? So of course, it's great to have such a diverse community, but also it's very hard to find any common ground. But I think that is a pure strength, right? So yeah, I'm optimistic, and I hope that in the future, right, we will try to maintain this connection, right, and try to find these points of intersection, and who knows, maybe for some future, maybe collaborations on some common, grant proposals or whatever, right, whatnot. So I think that would be very interesting. And of course, thanks to our audience, thanks to you, right, Michael, for coming here and delivering your presentation, and thanks to Ilya for making this happen, and thanks to all our great mathematicians in the Zoom room today, right, tonight, for being with us and for asking questions and for listening to us. And I believe that many more will watch this on this recording on YouTube, and let's keep on doing the right thing. So thank you so much. Thanks everyone, and let's have
Thank
a
you,
good
Dr.
one.
Tavrov, Ilya.
Yes.
Всі.
Thank
Goodbye.
you. Thank you.
Щиро вам дякую. До побачення.
So